Let me introduce the last speaker of this first session. Is Ennifar. Is a researcher of the French National Center of Scientific Research, and the title of his talk is Incremental ITC Reveals the Mechanism of Action of HIV-1 Reverse Transcriptase and Inhibition Mechanism by non-nucleoside inhibitor.
Hello, everybody, and thanks to the organizers for the invitation to this very nice place. So I will illustrate through two different examples how we use ITC in the lab, and especially an approach that we introduced some years ago, we termed Incremental ITC, to study molecular machines. And can you just please use the PowerPoint instead? Yeah. Okay, and so the first example is the HIV-1 reverse transcriptase, and I will show how using ITC we could reveal the mechanism of inhibition of this enzyme. So first I will start by saying some words about HIV-1 reverse transcriptase, RT. RT is a key enzyme in the viral life cycle, converting the single-stranded genomic RNA from the virus into a double-stranded proviral DNA. It's also an essential enzyme for anti-HIV-1 therapy because half of the compounds that are currently used are targeting the polymerization activity of RT. And RT inhibitors can be divided into two families. First, NNRTI, nucleotide analog RT inhibitors, that are competitive with the natural nucleotide substrate of the enzyme, but they act as chain terminator after incorporation into the primer strand. And the second family are NNRTI, for non-nucleotide analog RT inhibitor. They bind to a hydrophobic pocket located about 10 angstrom away from the catalytic active site. They are non-competitive inhibitors, but their mechanism of action was still unclear. So briefly, some words about the mechanism of action of RT. The proteins binds double-stranded nucleic acids, forming this protein nucleic acid complex. Then the incoming nucleotides bind to this protein nucleic acid complex, forming this open ternary complex. Then the incoming nucleotide is incorporated into the primer strand during the polymerization reaction, and an inorganic pyrophosphate is released. And finally, the protein translocates for the next round of polymerization. And our initial goal was to investigate this mechanism of action using ITC. And we started by the very beginning, the binding of protein to double-stranded DNA, primer template DNA. So this was investigated over a large range of temperature, between 5 and 30 degrees. And you can see that from 10 degrees and above, the binding of the protein to the DNA can be well described by a one-to-one binding site model. The protein binds as expected to the DNA, forming a catalytically competent protein-DNA complex. But at 5 degrees, the system is more complex, and we have to introduce, in fact, a second binding mode to analyze the data. And if the main normal binding mode is the correct one, what about this second binding mode? And I will come back on this later. So now if we plot, if we look at the temperature dependency of thermodynamic parameters, you can see that there is a strong dependency of the delta H. This is due to the heat capacity change, delta Cp, which is strongly negative in this case, which is in full agreement with the crystal structure of the free protein and the protein bound to DNA, showing that there are large protein refolding upon DNA binding. You can also see that if we extrapolate our data towards higher temperature, we expect a delta H equal to 0 around 34 degrees. And according to Van 't Hoff's relationship, we expect an extremum of the KD when delta H is equal to 0. And in this case, very interestingly, we have an optimal DNA binding affinity that occurs close to the physiological temperature for the virus. So now our objective was to follow RT during polymerization, RT in action. And for this, we introduced incremental ITC that allows to follow a successive chemical step in the same ITC experiment. So here is an example of such an incremental ITC experiment, where we start with a normal titration. The protein is placed in the sampled cell and the DNA in the syringe. And we made a titration, and we obtained thermodynamic parameters for DNA binding. But at the end of the reaction, we keep the product of the reaction in the sampled cell and reload the syringe with the second substrate of the reaction, which is the first incoming nucleotide. In this case, DTTP, but we use instead a chain terminator DDTTP. And we perform a second titration on the same sample. And we have, in this case, an exothermic signal and a second titration that provides thermodynamic parameters for the sum of two reactions, the binding of this incoming nucleotide and its polymerization into the primer strand. We see the sum of the two reactions. We cannot make distinction between these two reactions. And again, at the end of this reaction, we keep the product of this second reaction in the cell and reload the syringe with a third substrate of the reaction, which is a second incoming nucleotide, in this case DATP. And we perform the third titration on the same sample. And because at the previous step we use a chain terminator DDTTP, the second incoming nucleotide cannot be incorporated anymore into the primer strand. It can bind only. So with the third titration, we have access to thermodynamic parameters of the incoming nucleotide binding only in absence of the catalytic reaction. And the difference between these two reactions yields thermodynamic parameters for the polymerization reaction, the chemical step, in absence of binding. And this you cannot get by any other approach. So using this incremental ITC approach, we could follow up to 5 consecutive reactions on the same sample. So here is an example where we start by DNA binding to the protein. When we saturate the protein with DNA, we stop the titration. Then we add the first incoming nucleotide, DCTP, and we see binding and polymerization of the first nucleotide. We see then binding and polymerization of DTTP, binding and polymerization of the chain terminator DDATP, and finally binding only of DGTP on the same sample. And using this incremental ITC strategy at various temperatures and for all 4 natural nucleotides, we could obtain a complete dissection of DNA synthesis into multi-step component by the HIV-1 reverse transcriptase. Here I just show the result for DATP. So the curve we obtained for delta H, for T delta S and delta G. So we obtain parameters for polymerization, the overall reaction, the binding only, and the chemical step plus translocation. In fact, we cannot distinguish in our experiments between these 2 sub-steps. And what is interesting, if you focus on this graph, is that the polymerization reaction alone, the chemical step, is purely hexagonic, whereas the binding only is highly hexagonic. And this strongly suggests that it's the DNTP binding that provides most of the driving force for DNA polymerization, and not, as expected, the chemical step. And now let's come back on this strange data we obtained at 5 degrees. By the way, it's interesting to make this experiment at different temperatures, because I'm crystallographer. When you are crystallographer, you try to crystallize or to co-crystallize complexes, protein DNA complexes, at 2 temperatures, 20 degrees and 4 degrees. And by looking at this data, you can clearly see that at 20 degrees, we have one homogeneous complex. At 4 degrees, very likely, you have a heterogeneous complex. So don't do it at 4 degrees, at least in this case. So we know from our incremental ITC experiment that at 10 degrees we have 100% of our complex that is active, able to incorporate incoming nucleotide. At 5 degrees we have some second binding mode that is introduced with this primer template DNA sequence. But if we slightly change the sequence, at 10 degrees we already have two binding modes. And if we look at the thermodynamics of these two binding modes, we see that they are equally populated. One has a very tight binding in the low nanomolar range with a delta H about 13 kilocal per mole. And the second one has a KD around 83 nanomolar and a higher delta H plus 24 kilocal per mole. And we know that this low affinity corresponds to the thermodynamic signature of an active RT-DNA complex. So what about this very tight binding? Is it active or not? So to understand this, we perform incremental ITC experiment by the addition of the next incoming nucleotide into this sample, in this case the ATP, and we see binding and polymerization of the ATP. But we see binding and polymerization of only half of the population. So this active population is able to polymerize the ATP, not the other one. So clearly, this very tight binding corresponds to a catalytically incompetent binding mode. So now, what about non-nucleoside RT inhibitors, NNRTIs? A model was proposed for NNRTI inhibition in 1995, published in a science paper. And in this model, the protein binds the DNA. Then the inhibitor can bind to this protein DNA complex. And the presence of the inhibitor do not interfere with the NTP binding. Then we have this quaternary complex, in fact, with the protein the DNA, the inhibitor, and the incoming nucleotide that is bound, but the presence of the inhibitor blocks the chemical step, the polymerization reaction. So this was the model that was proposed, and it was almost a dogma for 20 years, almost 20 years. And all the pharmaceutical industries try to improve these inhibitors relying on this model. And as you know, it's important to know the model of inhibition when you want to improve an inhibitor. So we decided to look at this NNRTI inhibition using ITC. And we know that we can clearly see the binding of GNTP by ITC. For this, we introduce a chain terminator into the primer strand. And when we add the next incoming nucleotide, like here the ATP, we see binding only. It's highly exothermic. We have this very nice titration, in this case for the ATP. So we can clearly see the NTP binding only. So now, what about the effect of the inhibitor on DNTP binding? So in this case, we pre-incubate DNA, protein, and the inhibitor, and we add the next incoming nucleotide. So based on the previously published model, we should see something like this, binding only, because they claim that the inhibitor do not prevent the NTP binding. And by ITC, and at different temperatures, we just see this, nothing, no binding at all, which clearly means that the inhibitor, the incoming nucleotide, cannot bind the RT inhibitor DNA complex. And this is in complete contradiction with the model of inhibition. So based on these results, we decided to completely reinvestigate the model of inhibition. And we started by looking at the binding of the inhibitor to a pre-incubated protein DNA complex, in this case, nevirapine. And we have this kind of binding curve, so it binds. The affinity is not very high, but it binds. And now what about the activity of the protein after binding the inhibitor? So in an incremental ITC experiment, we had the next incoming nucleotide, the TTP, and we see this. So most of the protein is not able to bind. But there is a very small fraction that still do something. In fact, we could show that it binds and polymerizes the TTP, because in this experiment, the affinity of the inhibitor is so weak that there is a very small fraction of protein DNA complex that is still active. And we think that's very likely why in the previous model they used biochemical approaches. They made a mistake because they underestimate this very small fraction of active protein DNA complex. And you will also notice this very quite unusual shape of injection peak. In fact, the peaks are very broad. And as presented by Philippe, we use WIDEN-ITC software to retrieve kinetic information. So with WIDEN-ITC, we could get we have a complete thermodynamic and kinetic description of the binding of the inhibitor to the protein DNA complex. So not only KD, delta H, delta S, and delta G, but also K on and K off. And we could show that the inhibitor binds the RT-DNA complex very slowly. And we could also show that the affinity of the inhibitor for the protein DNA complex is only in the mid-micromolar range, which is not very good for an inhibitor, but okay, it's still working. And we confirm that the incoming nucleotide is not able to bind to the protein DNA inhibitor complex. Now next, what about the binding of the DNA to a pre-incubated protein DNA complex? So we use two kinds of inhibitors, nevirapine and efavirenz. And in this case, we see this kind of binding curves. And again, we see that the DNA binds the protein in two different, or the protein binds the DNA in two different modes. And one of these two binding modes involves a very tight binding. It's in the low nanomolar range for nevirapine and even in the picomolar range for efavirenz. So now, are these two binding modes active or not? So we perform incremental ITC experiment. On this sample, we add the next incoming nucleotide, TTTP, and we see that we have no binding at all. So both binding modes correspond to inactive complexes. So with this, we have shown that this, the presence of the inhibitor promotes a second binding mode of the free RT onto the DNA, and that this second binding mode has a strongly enhanced affinity towards DNA. And finally, we investigated the binding of the inhibitor to a ternary complex made with protein, DNA, and the incoming nucleotide that is bound. And in this case, we could show that the inhibitor cannot bind this ternary complex. So putting all this data together, we can now propose a new mechanism of inhibition for NNRTI, where we have to distinguish between two inhibition pathways. A first one where the protein binds DNA, forming the polymerization competent RT-DNA complex. The inhibitor can bind to this complex, but very slowly. But when it's bound, it prevents the binding of the incoming nucleotide, and the enzyme is inactivated. However, in the cell, there is a large concentration of DNTP. And DNTP binds very fast to the polymerization competent RT-DNA complex. And when it's bound, it prevents, as shown just before, the binding of the inhibitor. In addition, when it's bound, it's immediately incorporated into the primer strand in the polymerization reaction. So in other words, as soon as the protein binds the DNA and forms a polymerization competent RT-DNA complex, when the RT enters into the polymerization cycle, it cannot be stopped anymore by the inhibitor because of a kinetic barrier. So hopefully there is a second inhibition pathway where the inhibitor binds the free enzyme, and the presence of the inhibitor into the enzyme strongly enhance the affinity for the DNA, but promotes the binding in the wrong orientation. And the protein is trapped in this inactive dead-end complex that is very stable. And we think that's the way the inhibitors are working in vivo. Not this way, but this way. So using ITC, we have this quite complicated, but I think more accurate model of inhibition for inhibitors. And again, if you think that all the pharmaceutical companies worked for more than 15 years on the wrong inhibition model, I think it was really worth using ITC. And also to show the importance of ITC. So all these structures of RT bound to different inhibitors were known. There were many crystal structures that were solved. So the structural knowledge of the system was very good. But there was a clear lack of thermodynamic and kinetic data that were collected through keen ITC and incremental ITC to make a link between the structure and the thermodynamics and the kinetics. And it's only by collecting and gathering this structural and thermodynamic and kinetic data that you can clearly understand how the machine is working. So now RT is a nice system, but it's rather small. It's only one protein, one nucleic acid, one or two ligands. And we wanted to play with a more complicated system, so we decided to investigate the 30th translation initiation complex, the ribosome, which is more funny. So the ribosome, maybe you know, involves 4 different steps. The initiation, the elongation, the release of the nascent peptide, and the recycling of the ribosome. And we started by the investigation of the initiation of the translation, so the assembly of the translation initiation, which involves 30S small subunits, the large 50S ribosomal subunits, initiation factors 1, 2, and 3, and initiation factor 2 is bound to GTP. It involves AFFINimeter tRNA initiator and a messenger RNA. And all these protein and nucleic acids first bind to the small ribosomal subunits to form the 30S pre-initiation complex. Then the AFFINimeter tRNA established bonds with a messenger RNA, codon-anticodon interaction, to form the 30S initiation complex, which is then recognized by the large ribosomal subunit, which binds and ejects and releases initiation factors. And our goal was to see whether there is a preferred assembly pathway of this initiation complex. And for this, we tried to obtain a thermodynamic landscape of the whole bacterial 30S translation initiation assembly. So for this, we used our incremental ITC strategy, just here to illustrate. We start with a 30S in the ITC cell. We add IF3, and we have binding parameters for 30S IF3. Then we add another factor, and we have binding parameters for 30S IF3, IF1 binding, and so on and so on. We add messenger RNA. So using the same strategy that was developed previously for RT. So the first question is, what is the first partner binding to the small ribosomal subunit? Is it IF1, IF2 GTP, IF3, or messenger RNA? And for this, we selected on the basis of the affinity measured by ITC. Of course, the affinity is not the only selection criteria. It can be also the kinetics, for instance, or the concentration of each factor in the cell. So what is the first factor binding to the ITC? We perform all the experiments at different temperatures. You can see that for IF1, the delta H is close to 0 at 20 degrees, but it's not the case at different temperatures. That's why it's important to perform always the experiment at different temperatures. And we could see that IF3, for all the temperature range investigated in this study, had the best affinity, and it's the first partner that should bind to 30S. So IF3 is the first factor binding to the 30S. What is the next one? Is it IF1, IF2 GTP, or messenger RNA? So on the same sample that was formed before, we add either messenger RNA or IF1 or IF2 GTP. And in this case, here at 25, but it's the same at 20 or 30 degrees, IF1 is very likely the second binding factor, the second factor binding to the ribosome. And so on and so on. We can go on with this experiment using incremental ITC, and in this case it's important to recycle the sample because it's very sample consuming, several milligrams of ribosome for each experiment, so we can do many experiments on the same sample. So we could obtain this as a scheme of all the experiments that have been performed so far, or almost. There are more than this at the moment. With here the temperature at which all the experiments were performed. So for instance, here we start with 30S, we add IF3, then we add IF1, then we add messenger, then we add IF2. Or we can start with 30S, we add IF1, IF3, and so on. We have a complete map of interaction of all the factors. And I would like also to focus on this interesting interaction. The question was either an affinity variation of mRNA binding. In the literature, it was described that the binding of the messenger RNA is not dependent on initiation factors. So we can check using ITC. In absence of any factor on the free small ribosomal subunit, using this messenger RNA sequence, here in red is the Shine-Delgado sequence that is supposed to bind to the anti-Shine-Delgado sequence present in the 30S ribosomal subunit. So we add this, place this into the syringe and add this to the ribosome, and we obtain this thermodynamic parameters. So delta H is about minus 59 kilocalories per mole. And this is rather good agreement with what is expected for Shine-Delgado, anti-Shine-Delgado hybridization. So it's interesting to see this. And now if we add initiation factors, either IF1, IF2, IF3, or IF1, 2, and 3, you can see that there is a clear influence of the presence of the initiation factors on the binding, especially on IF3, which improves the binding. And you can see that the thermodynamic signature of the binding is dramatically changed compared to the free ribosome. So there is a strong influence, especially of the presence of IF3 on messenger RNA binding, and this was not described in the literature. And finally, when does the AFFINimeter tRNA initiator bind? So we could not observe any binding in absence of IF2 GTP. So here is shown the result for the binding of the tRNA to 30S IF2. And we have this kind of binding curve. But now if we do the same on the complex containing the ribosome, the messenger RNA, IF1, IF2, and IF3, we have a much better binding, about 4 times better. So clearly showing that there is a requirement of the presence of all factors on the ribosome for the tRNA to bind. And also, yes, to increase the affinity. And so if you look at these injection curves, you can see again a broadening of these injection curves, clearly, which is clearly the signature of a slow kinetics of binding. And using KIN-ITC, we could derive kinetic parameters for the binding of the tRNA, and we found data that are in good agreement with fluorescence data that were described in the literature. So very probably the tRNA binds slowly to this complex, and it binds at the end of the initiation. So all the data are not, the collection of the data is not finished yet, but this is the model we can propose at the moment, where 30S bind first IF3, then IF1, then we still have to place IF2 GTP somewhere. We have to make more experiments, we don't know yet. But then it looks like the messenger RNA binds to the ribosome, and finally at the end the tRNA binds to this complex, forming this 30S initiation complex. So with this I'm finished, and I would like, so first this work was done in our lab, Architecture et Réactivité de l'ARN in Strasbourg, in the group of Philippe Dumas, And I'd like to thank especially Benoît Meyer and Guillaume Beck, who have performed most of these experiments on the RT and on the ribosome. And I'd like to thank our collaborators from the University of Innsbruck, the Ronald Mikura group. They provide very important modified DNA that was used as a control experiment for RT. And our collaborators from IGBMC in Ilkirch, Marat Yusupov and Gula Yusupova, for advices for the ribosome. And thank you.
So we have time for a couple of questions. Yeah, I think in the chat.
Hi. This is a question. This is a question from Sarita Tripathi from. No? Hola. Oh, this is a question from Sarita Tripathi from Central Drug Research Institute in India. I hope it's right, the question. I have a question regarding the volume and concentration of sample cell in step titrations for the same sample with different ligands.
So you can perform this kind of incremental experiment provided there is no interaction between the substrate you use at one step with the substrate used in the previous step. It should not interfere with all the previous substrate. This you have to check. And about the concentration, of course, you have to start with a concentration that is sufficient to allow a correct titration in the subsequent steps, because you have dilution. And the more you do the experiment, the more your sample is diluted. So you have to start with the correct concentration. But it's the same than a normal titration. You just have to take care that you don't have interaction with a previous substrates.
Following the same question, but how do you perform this incremental experiment? So do you, in order to not overflow the cell, so do you remove the exit or just keep some room for the next?
We remove the excess and that's it.
My question? Yeah.
So actually following on exactly all those same questions, how many titrations are you confident you can do in a row before the small errors all add up and it stops working?
Good question. So of course we compared. We are confident because we compared. So if I come back at the very beginning. Here, if we compare, for instance, this kind of experiment here at the fourth step with a preformed complex, we have the same. So this means that it's, of course, provided your protein is stable. This kind of experiment we start very early in the morning and we finish quite late. And of course, to optimize your experiment, it's better to inject small volumes, or not too small, but not all the volume of the syringe, so you limit the dilution of your sample, and that's it.
Should just repeat that you were able to perform 5 successive titration in the same experiment.
Hello. I have a question on the Van 't Hoff equation that was mentioned here and before. Until what point can we trust delta H quantitatively if we don't use different buffers?
So in this case, the delta H that is shown is the interesting delta H of the reaction. We to care about any effect of the buffer, first by performing the experiment for RT-DNA in 3 different buffers, and we have the same delta H. And we like to use cacodylate, which has delta H of ionization very close to 0, so it prevents such problem.
When you do the successive titration, since you remove the excess, because otherwise it would be impossible, do you correct each time the concentration before?
Okay, we have no more questions. We thank again Eric and all the speakers in this morning.